Sir, are you there?
Yes, yes.
Okay. So Dr. Murthy sir, our director sir, is in some important meeting. He is engaged there. So right now he just convey the message that he is from Department of Pharmacology, Toxicology and Regulatory Toxicology Department on behalf of NIPER R fraternity. It is my pleasure to welcome all respected dignitaries, our director, sir, Dr. U.S.N. Murthy, our dean, Dr. Abha Sharma, our registrar, Dr. Jain Narayan, and all the faculties and colleagues, and especially all the participants who has joined today's webinar. Today, you must have heard a lot about the new technology which has come into existence nowadays. Yes, I am talking about the 3D printing and tissue engineering. The extraordinary minds at India and abroad, and I'm at MIT, they are working, and all over the world, they are working and have been the source of several 3D printer innovations over the years. And in the earlier webinar, you have seen that we, in the January that we have conducted in the Azadi Ka Amrut Utsav, we were focused on the regenerative medicine in Alzheimer's. In continuation with that, today's seminar we are focusing on the tissue engineering and organ printing. As tissue engineering is as also a subfield of regenerative medicine, and we will also focus on even more prevalent condition in which the restoration of functional tissue would answer the currently unmet need. So we welcome the today's speaker, Dr. P. Gopinath. His title of talk is Tissue Engineering and Organ Printing, and we will focus on all these issues which is related with the tissue engineering. In his talk, he will explain about the basis of tissue engineering and application of nanotechnology in tissue engineering. He will be also explaining about various bioprinter technique which is involved to print the artificial organs. So with this note, I invite Dr. P. Gopinath. I will also introduce a short biography of Dr. Gopinath. He is a professor in the Department of Bioscience and Bioengineering at IIT Roorkee. Currently, he is the head of the Center of Nanotechnology. He has done his MSc in biotechnology and then his PhD in biotechnology from IIT Guwahati. He has done his postdoctoral research at the University of Rochester Medical Center, New York, USA. His research group currently is working at the Nanobiotechnology Laboratory at IIT Roorkee on developing various nanomaterials for biomedical applications. At present, he has more than 120 research publications in the area of nanobiotechnology in a very high impact factor general. Even he has filed 10 patents and done one technology transfer. He has also published 8 books with 15 book chapters. He has been a reviewer and editor of various reputed journals, and his research work has been well cited. In total citation of the work is 4,546 and with H index of 35 and I-10 with 86 till date. He has completed 7 sponsored projects from different agencies like DBT, DST. Further, he has received several prestigious awards like ILSI India Young Scientist Award 2021, AMI Embellic Award 2020, ICMR Prize for Biomedical Research 2019, Shri Om Prakash Sharma Award, Young Scientist Award, and also the GD Naidu Award for Young Scientist Year 2018. Professor H.S. Sharma Award, Foundation Young Scientist Award. His credentials are very much impressive and recognized by the outstanding and recognized by our IIT Roorkee, and based on his contribution at the institute at IIT Roorkee, he received the prestigious Institute Research Fellowship, uh, Research Research Fellowship for Outstanding Young Faculty in 2017, IIT Roorkee. His research group developed nanofiber-based scaffold and device for tissue engineering application. For this work, his team also got the Birag Getty Award in 2019 and from Shri M. Venkatesh Naidu, Honorable Vice President of India. Dr. Gopinath also received the prestigious Virendranath Multi-Mittal Award in 2018, and many more awards are come into his bag. So I will just introduce few here. He is also the fellow of he's a Royal Society of Biology, Royal Society of Sciences, and Royal Society of Chemistry. So with this short introduction, I would like to invite our today's guest, Dr. P. Gopinath. Sir, over to you.
Yeah, thank you, Dr. Sabha, for the nice introduction, and also providing me this wonderful opportunity to discuss the topic tissue and organ printing at NIPER library. So virtually not able to come uh, in the offline mode to see all of you. And also, I'm happy to see your growth and success in NIPER library. And I'm seeing a lot of young colleagues are there. So in my talk, I'll be briefly talking about the tissue engineering and organ printing. So it will be a kind of lot of basics will be there. So I'll be explaining everything in a layman term. So I think the students can enjoy the talk. So I'll share the slides now.
Yes, sir. Yes, sir, it is visible.
Yeah, slides are visible, right? Okay.
Yes, yes.
So in my today's talk, that is
Sir,
tissue engineering.
will you please go to full mode, screen mode?
Yeah, it is in the full mode.
Now it is fine, sir.
Okay. So in my today's talk, like I'll be talking on the tissue engineering and organ printing. So I'll be covering the topic, what is tissue engineering and what are the applications of nanotechnology in the tissue engineering and organ printing and what kind of work we are doing in our lab at IIT Roorkee briefly. So before I go into the talk, like what is tissue engineering in details, let us see the brief statistics. So what is the need for the organs today? So if we see the statistics in the US alone, so in the US alone, at least 1,19,000 people need the organ transplantation. So they're waiting for the organ transplantation. And if you see that out of them, only 30,000 people are able to get the organ transplantation. And every day, at least 22 people are dying due to lack of suitable organ. So you can see the statistics. In US alone, 22 people are dying every day. You can think about the whole world. Okay, how many people are dying due to lack of suitable organ transplantation? So we can see the brief statistic about in India. Like, so in India, almost 1.5 lakh people need a kidney transplantation, but only 3,000 people are able to get the kidney transplantation, and remaining 90% of the people waiting list. So they are dying without getting the suitable organ. And similarly, in the case of liver, so we need at least 25,000 liver transplantation, but out of that, only 800 are able to get the liver transplants. And out of that 800 also, 70% are from the live donor, and the remaining 30% from the cadaver, that is the brain dead people. Okay. So this is a brief statistics of India. So if I ask you the cost of your mobile phone or if you ask you the cost of your bike, you can easily tell what is the cost. But if you ask you the cost of your organ, you won't be able to. So according to this website, this is the cost of each and every organ. You can see what is the cost of your liver, what is the cost of the lungs. Okay. So you can think about like how much important each and every organ. So in a lighter mode, I used to tell to my students like, don't ask your parents how much property you given to me. Okay, so your parents already given this much worth of property to you. Okay, so it's your job how you are going to use these and achieve success in your life. So jokes apart, and to move on to the topic, like what is tissue engineering? So the tissue engineering is a multidisciplinary field where we need the help of biological engineers, electrical engineers, electronics engineer, mechanical engineer. We need help from all the different discipline people so that we can make the complete organ which can restore or maintain or improve the tissue function. So why we have to make the artificial tissue in the lab? Because we don't have sufficient donor, okay, and the organs which are available also short in supply. So that is the main reason we are not getting proper donors, and the organs also short in supply. And we cannot take organ from any person. Okay. Jokes apart, so even though he is our close friend or close relative, like what is tissue, he cannot donate the organ. So the tissue is the organ should match with your body. Okay. So if you are taking any organ from other person, then our immune system will reject it. So we want to minimize the immune system, shorten supply. And how we can minimize it? We can take our own cells and we can create the artificial organ in the lab, and which we can take organ from any person to replace the diseased or damaged tissue. So that is the role of tissue engineering. So for the tissue engineering, what we are going to do, we are going to take the patient's own cell, okay, and we are going to add it on the scaffold. Scaffold means support. For example, if you want to construct a multi-story building, you need the foundation, okay, and you put a different kind of foundation for the multi-story building, and you put a different kind of foundation for the normal simple home, okay. So depends on the organ, we have to select the proper scaffold, and the scaffold should be porous in nature so that the cells can attach and also the nutrients can easily pass through, okay. So this is the overview of tissue engineering. The first step is
Pharmacology,
we can
Toxicology
remove
and
the
Regulatory
cells from the
Toxicology Department
patient's
on
own body,
behalf of
and the cells can be grown in the two-dimensional cell culture plate or cell culture flask or cell culture dish. So once we get the sufficient number of cells, then we can add the cells to the three-dimensional scaffold, and the cells will grow on the three-dimensional scaffold, and once the tissue is completely formed, then we can replant the engineered tissue in the damaged site. So this is the overview of tissue engineering. So why we need three-dimensional scaffold? So usually in the lab we grow the cells in the two-dimensional glass plates or the cell culture dish. So where the cells will attach and grow in the 2D. But inside our body, if you see the in vivo, so all the organs are 3D. So to mimic our original environment, so we have to use the three-dimensional scaffold so that you can transplant the organ to the particular person or the particular animal. So what are the three important tools of the tissue engineering? The first thing is cells. The cells are the living part of the tissue. So the cells, it can be a stem cells or it can be the progenitor cells. We have to select the right cells, and next one is that we have to select the right scaffold. So depends on the organ, we have to select the right scaffold. The important property of the scaffold should be it should be a biodegradable and biocompatible. So what is the difference between biocompatible and biodegradable? So biocompatible means it should be compatible to your body, and biodegradable means once it is degrade inside the body, because after some time the scaffold will degrade and our original tissue will replace the artificial tissue after some time. So when the scaffold degrades, the degraded product should not induce any toxic effect. So that is called as biodegradable property of the material. And third important property is cell signaling. Okay, so you have the stem cells, and the stem cells don't know whether it has to differentiate become liver cells or it has to differentiate into kidney cells. And this cell signaling plays an important role in instructing the stem cells to become a liver cells or the kidney cells. So these are the three important things required for the tissue engineering. So as I told you, the first thing is the cell source. So it can be a adult stem cells or it can be embryonic stem cells or progenitor cell. So one of the problem with the embryonic stem cell is you have to get the stem cells from the embryo, right? So we need an aborted fetus. So that's why most of the country, due to ethical reason, this embryonic stem cell research is banned. And we can use the progenitor cells, but again, when compared to the progenitor cells, the adult stem cells are less ethical and it has a kind of optimized protocols. So we can use these adult stem cells for various applications. So we can take it from the bone marrow and we can grow the cells, okay, and we'll get the adult stem cells. And from these stem cells, we can take it from the patient body and we can grow in the lab condition and we can make the organ or artificial tissues. And next thing is the scaffold. So ECA means extracellular matrix. So depends on the organ, you have to select the right scaffold. For example, if you want to replace the knee joint or hip joint, where our body has it has to withstand more amount of weight. To withstand more amount of weight, we can use the metal scaffold. And in case of some of the small bone damages, we can use the ceramic scaffold. In some cases, we have to use the synthetic polymer. For example, if we want to make the artificial blood vessels, so we don't want the biodegradable. So we want the synthetic polymer which can stay for at least 15 years or 20 years. And some cases we can use the natural polymer. For example, you are making the artificial skin. So after some time, the artificial skin will be replaced by our original skin. So in those cases, we can use the natural polymer which can biodegrade after some time. So the very important factor is the cell signal. So you have the stem cells. As I told you earlier, the stem cells don't know whether it has to differentiate into liver or kidney. Okay, for example, I'm telling. So how these stem cells know it has to become as a liver cell or it has to become as a kidney cell? So we have to give the signal. So how we can give the signal? We can add the growth factors, or we can add some kind of hormones or the drugs. Then the stem cell will differentiate into liver or kidney cells or heart cells, whatever it is. Okay. So we can differentiate the stem cell into the particular organ type cells by providing the growth factor signal. So in a simple layman term, so we are going to provide the environment, so which will support the growth of the particular type of cell. So you might have seen in the old movie, okay, the twin brothers who travel in that train, and during the train journey they get separated. And out of the twins, one will grow in the villain family and one will grow in the hero family. So the one who grow in the hero family, he became a hero, and the one who grow in the villain family, he became a villain. Even though they are identical twins, okay, and depends on the environment, one became a hero and one became a villain. Similarly, the stem cells depends on the environment. The stem cell can become as a liver cell or it can become a kidney cell. Okay, it can may depend on the environment or the growth factors, whatever you give. According to that, the stem cells can differentiate. So what is the role of this mechanical force? So the mechanical force is also very, very important. For example, you might have also observed the boys who stays in the same hostel and eating the same mess food. Okay, and the person who goes to gym, he get very good muscular development. Okay, the person doesn't go to the gym, so he don't get any muscular development. So he is giving the mechanical force to the tissues. So when you apply the mechanical force to the tissues, and the mechanical force will be converted into the chemical forces, that is the gene expression, then it will go into the protein expression. Okay, so then muscle development will happen to the whenever he do the exercise, so he get the muscular development. So that's why the mechanical force also plays a very, very important role in developing the tissue. So I'll just briefly go through the what is stem cells. The stem cells which can self-renew, that means it can replicate on its own, and also it can differentiate into different types of cells. So that is the advantage of the stem cell. In a simple layman term, the stem cell we can compare to the seed. When you put a seed, you get the plant. From the plant, you get the flower. From the flower, you get the fruits. And the fruits will make again thousands of seeds. Similar to that, from the stem cell, we can get the stem cells. And we can also differentiate into the particular type of cell, like liver, kidney, or whatever the organ. Okay. So that is the advantage of the stem cell. So it can be broadly divided into totipotent or pluripotent and unipotent. Totipotent means so it can give rise to any kind of cells. So the example is embryonic stem cell. And multipotent means it can give rise to multiple types of organs. For example, mesenchymal stem cell. And unipotent means it is a each and every organ have particular type of stem cells. For example, liver will be having a liver stem cells, kidney will be having kidney stem cells, and that can rise only as a liver or only as a kidney. Only one type, single type. Unipotent means single type. So you can see here, these are the specialized liver cell, specialized stem cell. These are unipotent stem cell. So these, the unipotent stem cell from the liver, it can only become as a liver type cells. It cannot be become a kidney cell. And multipotent cells, it can be a kind of multiple types. And totipotent, so we are going to take the cells from the blastocyst, so it can give rise to any kind of cells. Next thing is scaffold. So you have to make the scaffold, three-dimensional scaffold, and it should encourage the cell attachment and cell migration. And also it should be porous in nature, so that it can allow the nutrients, and also it can remove the waste material from your scaffold. So these are the various types of scaffold. You can see these are the porous and fibrous. So depends on the application, we can select the right scaffold. And these are some of the steps in the tissue engineering. Like once you get the biocompatible scaffold, you can add the cells, and the cells will multiply, and it form the final human tissue. Okay. And these are some of the organs which most of the lab achieved success. For example, skin, bone, and cartilage. So it is already successfully developed in most of the lab. Okay. And but these are some of the organs which are very complicated in making in the lab condition. For example, liver, heart, and lung. So one of the challenges, you can see that heart and lungs, it need a scaffold which can shrink and expand. That is one of the challenge. And also, if you see the heart and liver and kidney, so it have multiple types of cell and lot of blood veins. So that is also the major challenge, which I'll discuss later in detail. So let us see the skin tissue engineering. So during your childhood, you might have got a wound, and if you get a if the wound size is smaller, then you don't see any scar formation. It will be automatically sealed. But if the wound size is bigger, what will happen? It takes more time for uh, getting cured. At the same time, you can also see the scar formation. Okay, so this scar become identification mark in your birth certificate as well as your JE and all the certificates. The scar become identification mark, right? And what is the problem? Like why we are getting this scar? So you can see here. For assume this is like a 5 centimeter or okay, you got a big wound. So this much amount of tissue is lost. So and this both the sides of tissue it will try to come close and it try to seal the gap, but it lost more amount of tissues. So that's why it is not able to repair it completely. So you'll get the scar formation. But if you replace the same amount of tissue, okay, so then there is no scar formation. It repairs completely. It will seal it, and there is no scar formation. So how to repair the skin? There are two methods, in vitro and in vivo. So in vitro method, we can make the complete artificial skin in the lab and you can place it into the human body. And in vivo sense is you can put the scaffold in the damaged site. You can also inject the cells and the cells will grow in the patient body and it can replace the complete skin. So you can see this is a mouse model where you can make a small wound, and when you put an active scaffold and there is no scar formation, okay, and it is blocking the contraction and no scar formation. But in case of inactive scaffold, you can see due to the contraction you can see the scar formation. So what is the role of nanotechnology? Why we need nanotechnology in the field of tissue engineering? So initially researchers used micropore scaffold and microfiber scaffold. So what is the drawback of this? If you see the size of cells, okay, so if you are using the micropore scaffold and the size of the cells also in the range of micrometer. So when you use the micrometer size scaffold and the cells are also in the range of micrometer, so it is attaching only to the support area and there are a lot of gaps, so which does not mimic like original tissue. The same thing happens in the microfiber scaffold also. So there are a lot of gaps which does not mimic like original tissue. So if you see our skin, okay, or any tissue under the microscope, so you'll be having a nanofibrous scaffold, okay, that is called as ECM, extracellular matrix. So you have the nanofiber scaffold, so that is called as ECM, and on the top of that you are having micrometer size cells. Okay, the cells are in the range of micrometer, and the fiber is in the range of nanometer. So the advantage is that here the single cell can contact thousands of nanofibers. So the transmission of signals are possible, and which can closely mimic like your original ECM, that is extracellular matrix. Okay, that is the scaffold, that is the support. Okay, so you cannot see the foundation of the building. Okay, so we can see only the 5-story building. So the foundation is lying beneath. Similarly, you are not able to see the nanofiber scaffold, and the cells are attached, and it's making the final tissue. So this is how the extracellular matrix look like. So it is a combination of different protein, collagen and proteoglycan, laminin. So you can see this is a scanning electron microscope picture. This is a single cell. Okay. So here the single cell is contacting thousands of nanofibers. So when the single cell contacts thousands of nanofibers, so the transmission of fine signals are easily achievable. So that is the advantage. So how to make these nanofibers? There are 3 methods. First one is self-assembly, second one is phase separation, and third one is electrospinning. So I'm going to talk about the electrospinning, which is the simplest method and most widely accepted. And in our lab also we are using this electrospinning technique to make these nanofiber scaffold. So what is the principle of this electrospinning? So by using this electrospinning, we can make the nanofiber. So the principle of the electrospinning is similar to the cotton candy. So most of you might have seen this cotton candy machine during your childhood. So what he do, he add the sugar. Okay, he add the sugar crystals to this area, this hole. Okay, and when you switch on the machine, the sugar will melt and it become a viscous fluid. And due to the electrostatic charge, and it will form like a fiber. And the fiber he collected using a small stick. Okay, basically what we are eating is a kind of microfiber. Okay, and he add the pink colors as to attractive. And here, instead of adding the sugar, we are going to add the FDA approved biopolymer. So we can use any kind of polymer, but preferably if it is FDA approved, it is very good for translating the research. So we can use the polymer, and we can use the, instead of pink color, we are going to add the FDA approved drug. Okay, so once you add the drug, what happens is, so when you apply the electric current, so the droplet will come out of the syringe, and it will take after, based on the electrostatic charge, so it takes a shape like a cone shape. So this is a cone shape that is called as Taylor cone, name of the scientist. Okay, so after that, this the fibers will be pulled out and it will be collected in the collector. So the distance between the syringe and the collector is very, very important, and also the viscosity of the fluid, the solution also very important. So this is called a stationary collector. So when you use the stationary collector, you get the random fiber, and this is another collector called rotating drum collector. So when you use the rotating drum collector, so this is like a third bundle. So your nanofibers can be coiled around it. Okay. So when you use the rotating drum collector, you get aligned fibers. So you get this kind of aligned fibers. And when you use the stationary collector, you get this kind of random fibers. So it depends on the application. You have to select the random fibers or the aligned fibers. So accordingly, you have to select the particular collectors. And once you made the nanofiber based membrane, so you have to check whether this membrane is hydrophilic in nature. So why it should be hydrophilic in nature? Because when you put the, if you suppose we made a bio bandage or artificial scaffold, so our body is full of water. So it has to go like the membrane should go well along with our body condition, right? So when you put the membrane, if it is a hydrophobic in nature, like a plastic sheet, it is not going to stick it to our wound. Okay, so it should be hydrophilic in nature so that it can easily stick it to the wound site or the site where we need the artificial skin. And how we can check the property, hydrophilic or hydrophobic nature of the membrane? So there's a simple experiment called contact angle measurement system. So by measuring the contact angle, we can say whether the particular membrane, the nanofiber membrane is scaffold is hydrophilic or hydrophobic in nature. So when you put a drop of water and take a photograph using the contact angle measurement system, and by using the software, we can measure the contact angle. So if the angle is less than 90 degree, your material is hydrophilic in nature. If the angle is more than 90, it is hydrophobic in nature. If it is more than 150, your material is super hydrophobic in nature. So the simple layman example is when you put a drop of water on the plastic sheet, it will be intact. So it will be obviously super hydrophobic. So when you put a drop of water on the paper, okay, so the water will be absorbed, so the contact angle will be less. Okay, so that means it is a hydrophilic in nature. So this nanofiber based scaffold, not only for the biomedical applications, we can also use it for the edible applications. So you might have heard about the test tube burger. So where we don't have to kill the animals, we don't have to kill the animals to get the meat. So we can grow the meat in the lab. Okay. So recently Singapore has approved that, and we can grow the meat in the lab, and it can be useful for the consumers. So this is the article in the Nature from 2010, from pig to plate. Okay. So you can see here the scientists, they have taken the small biopsy from the pig, and they grown the tissue in the lab condition. And as I told you earlier in the cell signals, okay, if you are growing the cells in the normal three-dimensional scaffold, it is not going to mimic, or it is not going to taste like the original meat. So if it has to taste like a original meat, so it has to undergo all the mechanical forces, undergo mechanical pressure and everything. Okay, for example, if the meat has to taste like the leg piece, okay, so it has to undergo the whatever the mechanical load or the mechanical pressure the legs will undergo of the particular animal. So what you can do, we can also exercise the muscle. So we can artificially do the exercise, the muscle in the lab environment. We can stretch the muscles, okay, and based on that, it will boost up the protein. And we can also add the flavors, iron or vitamins, okay, and you get the meat which is ready to cook and eat. Okay, so from pig to plate. So that means like the nanofibers scaffold or the tissue not only for the biomedical application, it can be also useful for the edible application. So let us move on to the next topic, that is organ printing. So there are some challenges, as I told you, in the tissue engineering. For example, making the organ to the next level. Okay, that is a complicated process in case of the nanofiber based scaffold. So where we have to go for the alternate new technology, that is the organ printing. So this is the 3D printing technology, and by using that we can create the artificial organ. So it is similar to your inkjet printer. So in the inkjet printer, you'll be putting the color ink. So instead of that, you are going to use the Bioink. So Bioink is your cells. So your cells will be mixed with some kind of proteins, and that is called as Bioink. And instead of using the paper, okay, we are going to use the bio paper. Okay, so the bio paper is like a kind of gel. It's a kind of scaffold, and it can be a temperature sensitive or it can be a pH sensitive. Okay. So I'll explain you what is the advantage of that. So this is the conceptual bioprinter. So this is what people so that you will get a complete idea about the particular organ. And you have to select the design approach, whether you want to follow the biomimicry approach, self-assembly, and you have to select the suitable polymer. You want to go with the synthetic polymer or natural polymer, and what kind of stem cells you are going to use, whether you are going to use the multipotent stem cells or the pluripotent stem cells, and what kind of bioprinter you are going to select, whether inkjet or micro extrusion or laser. These are three types of bioprinters available, which I'll explain detail later. So once the organ is printed, we have to check it in a bioreactor whether the organ is suitable enough to implant. Okay, so whether if you are making an artificial heart, the heart should be able to pump. Okay, it should be able to efficiently work. So then we can go for the organ transplantation. So these are the same steps whatever I explained in the previous slide. So these are the three important steps in the organ printing: pre-processing, processing, and post-processing. In the pre-processing, so you'll be doing the imaging, and by using the CAD software, you can blueprint the particular organ. And next thing is, with the help of Bioink and Biopaper, and with the help of 3D bioprinter, we can print the particular organ. And the third step is processing, post-processing. Okay, once the organ is printed, we have to check it in a bioreactor whether the organ is able to work efficiently. Then only we can transplant to the particular patient body. So in the first step, that is the pre-processing, what you can do is we can do the imaging of the organ. For example, if you want to make an artificial heart, you can do the bioimaging, and using the CAD software, you can make the blueprint of the particular organ. And the next step is the processing. Okay, you need the bioink and biopaper. So how it can be printed? As I told you, we can use the biopaper, and this biopaper is like a kind of a matrix. Okay, so in a simple layman term, you can see that it's like a kind of roof of the building. So in the roof of the building, like you'll be initially they use the wooden sticks, okay, wooden and bamboo support to support the roof. Once the roof is set, they will remove the wooden support. It is similar to that. Okay, so we'll be using this bio paper. On the top of that, the bio ink, this orange color, the cells will be printed like this, like dot dot. And after some time, the cells will be fused together. So the cells will be fused together and form like a ring. So on the top of that, I again put another paper. So we'll be stacking one by one. Above that, we'll be putting a bio paper and bio ink. So we'll be printing a layer by layer. So once it is printed, and as I told you, you can use the temperature sensitive or pH sensitive bio paper. So you can say slightly adjust the pH or slightly adjust the temperature. So the bio paper will be removed, and finally what you get is this kind of tissue. So you can see here, this is a thing like how the bio printer works. So layer by layer, you can print the particular organ or the tissue. So you can these are called as bio ink spheroids. So the cell with the clump like a droplet, these are called as spheroids, and these are called as bio ink. It's a combination of cells plus some kind of collagen and proteins. And when you print it on the bio paper, so it can be a thermo-sensitive gel, okay, thermo-reversible gel. So when you slightly change the temperature, it become a gel, and change it to another temperature, it can be removed. Okay. And next thing is the bio printers. There are three types of bio printer. Before that, we'll see what are the types of bio ink. So we can use the collagen or alginate. These are some of the examples of bio ink. So the collagen is widely used, so that mimic your ECM, extracellular matrix, because of excellent biocompatibility, and also what is it like? It is a easy chemical modification. So most of the lab they use the collagen and alginate. And these are some of the example of biopaper. We can use the temperature sensitive, thermosensitive agarose, or we can use the pH sensitive biopaper. Okay, and these are the types of bioprinters: inkjet, extrusion, and laser assisted. So depends on the organ and depends on the advantage of each and every 3D bioprinter. We can select the right bioprinter. For example, in case of inkjet printer, it is similar to your inkjet normal printer. Okay, so where you can see here that we will be using the thermal or piezoelectric force to print the particular organ. So you can make these small miniature structure of the heart. Okay, so for the next week, Valentine's Day, you can print the heart and you can gift it. So these small organs can be printed. Okay, so each will be having a different kind of bio ink, and one can be adding the blood vessels, and one can add the cells, and it can make the final organ. And the another one is extrusion printer. So in the extrusion printer, it can be a pneumatic or it can be piston or screw type. So this is like kind of you'll be extruding through the small nozzle. Okay, similar to your toothpaste. Like, so you can make the scaffold like this. If you want to make the artificial ear, so you can make it in the scaffold. Then you can add the cells, and you can get the artificial ear. Okay, and when you add the cell culture medium, and it can grow in the lab condition. And third one is laser assisted printer, and it is similar to a laser printer. So here the laser pulse will dictate. So this is a cartridge, so which is filled with the cells, and depends on the laser pulse. So the laser pulse can go and give the shape. So it can give the heart shape, or it can give the square, or it can give the triangle shape. So according to the laser pulse, so the droplet will form, and the droplet will fuse the cells. And you can see here comparison between the three types of bioprinters, and the cell viability is more in case of laser assisted printer, but the cost is higher. And in case of inkjet, the cell viability is good and the cost is low. And in case of micro extrusion, the cell viability is less and the cost is also medium. So it depends on the application and depends on the, you have to select the right printer for your right application. And the post processing is the third step. Like once you printed the organ, we have to make sure that organ is properly working in your body. So once you printed the cells has to fuse. After several days, it has to fuse and it form like a final tissue. Okay, and once it is formed the complete artificial heart, so you have to keep in a reactor and you have to check it whether it is able to pump and work like a real heart. Okay, if it is not able to pump and it is not able to withstand the mechanical pressure, okay, then it is not useful, right? So we have to check it before we transplant it. So the recent research shows that a group of scientists, they made the small spider web like membrane, and this is a kind of like artificial pacemaker. So this could replace the pacemaker, and this can be placed on the heart, and it can make the heart to beat. Okay, and they tried in the rabbit, and in the rabbit when they removed the heart and they put this kind of membrane with heart, and it's able to pump and work like a original heart. So what are the challenges? Why the organ printing is not successful, even though it was started in 2011, and Dr. Anthony Atla, he is the father of organ printing. So he was able to print the organ kidney in within 7 hours, but it is not functional in humans. So what is the reason? The reason is it's very difficult to create the blood vessels between the tissue layers, because and also these some of the organs have a very specialized function. It's very difficult to replicate. So you can see the kidney have a lot of blood vessels and a lot of types of cells. So for the blood vessels, this group of scientists, they come up with the idea that we can use the sugar template. So we can use the sugar and we can make the artificial blood vessels. And once it is printed, we can remove the sugar template by dissolving it. But again, it is not successful. So still a lot of research is going on in making the organ printing. That's what, so this is one of the major challenge in case of organ printing. And this person totally come up with a totally new idea. So instead of printing the organ, so why don't we use the old organ from the dead body? It's like a similar to the refurbished home. So instead of destroying the old home and make a new home, so why don't we refurbish the old home and make it like a new home? So we can take the organ from the dead person and we can inject with the detergents. So what happens is the detergents will destroy all the cells of the particular person, and whatever left over is only the scaffold. So you'll get this kind of scaffold. So in this scaffold, we can inject the patient's stem cells. Okay, and this we can grow in the incubator. So when you inject the patient's stem cells, and which and we can grow in the incubator, and it can become like a similar to the original heart of the patient. And then we can use that the spider web membrane, and which can give the electrical stimulation. It can make the heart to pump, and it makes the heart to beat like a original heart. And this is another group of scientists. They use the spinach leaf. The spinach leaf at 0 day it is like this, and 7th day after using the treatment with the detergent. So using the detergent, so what you get is only this kind of scaffold, and this scaffold also look like a kind of blood veins, like it has the plant veins. It can also similar to your blood veins. So we can culture the heart cells on the top of this leaf-based scaffold. And the advantage is like we can bypass the organ donor list, and initially the cost will be high. And as I told you, there are a lot of limitations and challenges in making the blood vessels and different types of cells. So these are the challenges, but in future we can achieve it by making the complete organ with the less cost. Okay. So with this, I'll go on to the next 2-3 minutes. I'll discuss about what kind of work we are doing in our lab. So our lab at Nanobiotechnology at IIT Roorkee. So we are mainly working on biomedical applications of various nanomaterials. And in today's talk, I'll be focusing only on the tissue engineering application. So we recently made a 4-dimensional scaffold. So as I told you, there are some drawbacks with the 2D cell culture. So when we grow the cells in the 2D, so which is not going to mimic your 3D environment. So in the 3D environment also, these scaffolds are static. The 3D scaffolds are static. These are not dynamic. If you want to make an artificial heart or artificial lung, the scaffold should shrink and expand. So to make the scaffold to shrink and expand, so what we can do is we need an alternate scaffold. So to overcome that drawback, we made a nanofiber-based scaffold, multi-layer scaffold, and both the sides we added the magnetic nanoparticles. So when we add the magnetic nanoparticles, with the help of external magnetic field, so these scaffolds can shrink and expand. Okay, the scaffolds can shrink and expand, and you can see here the cycle 1, what is the expression, and the cycle 3. So based on the mechanical stress, the amount of protein expression will also vary. So you can see here this is a cell structure, and whenever you give the mechanical stress more to the cell, the stress fibers will be expressed, and the cells will also have a finger-like structure that is called as phylopodium. So that will try to hold the substrate where it is attached and growing. So you can see here from this picture you can clearly understand, like during the roller coaster ride we hold our sister hand or mother hand tightly. So similarly, so this is a cell, and these are the nanofiber based scaffold. These are the aligned nanofiber scaffold, and when we give the mechanical stimuli, so this cells are extruding that phylopodium, and it is grabbing this nanofiber scaffold. So there is a displacement of 5 to 10 micrometer displacement. It is grabbing the nanofiber scaffold, aligned nanofiber scaffold. Okay. And for this work, this was also, we filed a patent, and this also done a technology transfer, and it was the cover page of the particular issue by the editor. It was selected, and for this we got a Bioink Gandhi Young Technology Innovation Award from Vice President of India. And another work what we have done is we made a small nanofiber based cell culture insert. So the commercial cell culture inserts, these are slightly expensive. Starting from 500, it can go up to 5,000 rupees. So we made such kind of cell culture insert by simply using the pipette tips. Okay, so this is very important. The cell culture inserts are very important for understanding the wound healing studies. Like for example, you can see here the cells are moving from here, and also we can use it for the cancer metastasis. Cell culture inserts are very useful. So we have taken the micropipette tips and we cut it and we deposited the nanofiber, and we can use as a kind of cell culture insert. And if you see the cost, this is less than 1 rupee. Okay, and again the another additional advantage is we can completely autoclave. We can sterilize it by autoclaving method. So that is the advantage of this nanofiber based cell culture insert. And this work also selected for the cover page of the issue. So with this slide, I'll conclude my talk. And also I'm teaching a NPTEL course in like biomedical nanotechnology, on the topic biomedical nanotechnology. So every June month the course will be running. So if anyone is interested, you can register in the coming June month. Okay, every June month the course will be on. And this course not only for students, it will be also useful for the faculty because this is already approved by the AACT for the faculty development program. So those who are interested, you can register for this course in the NPTEL portal. The course name is Biomedical Nanotechnology. So this is my group, and I thank all my students and all the funding agency. And I am also happy to see all my students are joining in a lot of prestigious institute. For example, Dr. Sabha, she is doing a very good job in NIPER, I believe. And recently, my another student, Dr. Uday Kumar, he joined in IIT Tripadi, and so he's in CSO Chandigarh, and she is also in CSO Chandigarh. So, and Dr. Chokalingam, he is in NIT Warangal. So, I'm happy to see that all the students are getting into good position, and they are giving a tough competition to me also. So most of the time when I apply for a grant, they also I'm also able to see their name in the grant presentation. So that's a good, healthy competition. So thank you all. So thank you all for your patience and listening my lecture. If you have any queries or doubts, I'll be happy to answer your questions.
Thank you, Dr. I'm very happy that after a long time I'm able to see your lectures, and I'm always very impressed with Sir style, as he has given a very good example how he connect the work with the student also. How much important we are, the organs are, how much costly we are. So thank you for realizing, and I think students are getting very much uh, good information, and you can see the innovation what we are doing in India. So in NIPER also we can do all these kind of work. So if any questions are there, I'm inviting all the questions. Question is and session is open. One student has asked some question. Himanshu. Sir is not audible. No. Some people in the chat. Is any question is there? Please ask.
Yeah, any question you can type in the chat box if you are not able to unmute yourself.
Okay. I think they're all are muted. Okay. Sir, as you told me, I've asked a very small question to you, right? You told me it is very difficult. That is a long journey to go. There is still many things I know. But right now, as 10 years, 2011, this organ printing has come. So how much time do you expect that it will come into the market, or it will benefit for the human? It will benefit human in terms
Uh,
of cost also. For the common man, it will be available.
yeah, maybe it may take additional another 10 years because like if you see that for even for the artificial meat, so it took almost more than 15 years to get approval, and only one country now, Singapore, only last year they approved that due to the COVID, they're not able to export the meat from the other countries, so they given a kind of approval. So that's why like and if it is for the going into the human body, then again it need lot of other clinical studies. Okay, and recently last month, and I think they made a xenotransplantation from the pig heart. They made with a completely genetic engineering modification, and they made the completely humanized heart from the pig. And it was the first surgery. So I think based on the success, hopefully soon we may get such kind of trials, and we may get the organs.
Okay.
Maybe another
It
5 years or
also
10 years.
required, it also required some regulatory. It also go for regulation, like stem cells also require some regulatory requirements are there. They have to follow like clinical trials. You have to clear it, animals trial. You have to go for the IAC, and so this is also required for the organ printing. If somebody has prepared the organ for any regulations are there for this.
Yeah. So we have to go through like similar to the same like drug discovery to go with FDA approval. Without FDA approval, we cannot go for any for commercialization or any clinical trials. The same thing with the organs also. There are several international regulatory agencies are there, which we have to go through to take it for the translation level. And there are some questions in the chat box like they are not able to connect. I think Himanshu.
Sir, I have one question.
Ah, yes, ma'am.
Hello.
Ah, yes, madam.
Sir, in spite of the spinach, we can use any plant for that purpose?
Similar to the spinach leaf, IIT Kanpur, there's another professor, Professor Shiva. So
Mm.
he is using this paper rose plant.
Okay. Okay.
He's using that leaf of the paper rose. So anything, even in one of the country like Brazil, they use the fish skin. So the tilapia fish skin they used as a kind of skin for burn wounds. So we have to select the right scaffold and try it and justify it.
Okay. Thank you, sir. Thank you, sir.
There's one question in chat box that it is a wonderful presentation, sir. I have a query related to vasculature. Does this bi-layered or multi-layered scaffold favor in vascular modeling, or how can be it achieved? Please share some knowledge or light on this, sir.
Right. So it's a very good question. So the multi-dimensional scaffold, we can put the kind of blood vessels. So we have the flexibility to add that. But only thing is like how much it will closely mimic our original organ and how much can be withstand the the blood flow. For example, the heart. So it has to have a lot of pressure, right? The blood flow has to go in and out. So whether it can withstand the particular mechanical pressure, that is also one of the important challenge. So that we have to optimize all the parameters, but we are not working in the particular direction, but it is possible. We can still make it. So we have to put the right combination of the polymer, and we have to use such kind of blood vessels which can withstand the mechanical load and the blood flow. So that is doable.
So, any other question? All questions are welcome by the student. Maybe they are different kind of presentation, but pharmaceutics people are doing, I think, a 3D printing here. So they can ask question also if they have only. So,
Yeah, if you're not able to unmute yourself, you can type in the chat box.
you can type in the chat box also if you want. Maybe they have something. I think in between, because we have also lost some connections. So maybe
Oh,
the connection is lost by someone. Anyway, sir, thank you very much, and each and everyone, and especially our speaker, Dr. Gopinath sir, our director sir, and each and every one of you for being here with us today, especially to all the student and participant who has taken all my colleagues and speakers. I hope this webinar must open the new avenues as well as your zeal towards the 3D printing, because it's a new and very challenging field. So you have a lot to do here. So, and it is also open the new like translational things. When you are doing the basic science, you may not get some translation, but if you are going to the 3D printing in future, you have a lot of time also. I'm talking about also the student that you have the 20, 30 years ahead. So you can go do some translation also with these new ideas. So few of the students are writing, thank you sir for the uh, great information they are writing. So thank you everyone, the feedback is there. Every students are very much happy. Again, thank you, Ms. Sir, for accepting my invitation of coming here from your busy schedule. And I'm also thanking our director, sir. I hope that this pandemic will end soon and we will meet in person in 2021 only. So, because it's a long time that I have not seen you due to this pandemic. And I'm really very happy today that I get an honor that to call my one of my most important mentor. I learned a lot from him actually, and he always give strength to me ki, yes, you apply and you will get the grant, yes, you apply, you